Right, everyone. Good morning from Los Angeles. The sky is great, but it's very nice. My name is Alexandra Lieben. I'm president of ASciNA, a large independent network of Austrian scientists and scholars in North America. Our speaker today is Christina Baumgartner. She's a senior principal research scientist in the Immuno-Oncology Discovery Research Group at AbbVie, Chicago, Illinois. Tina received her master's in genetics from the University of Vienna and her PhD in immunology from TU Wien. She pursued postdoctoral training at Baxter BioScience in Austria, which included research at the Scripps Research Institute in La Jolla, California. She furthered her training at the Blood Research Institute in Milwaukee, Wisconsin. And prior to joining AbbVie in 2016, Tina held a research scientist position at the Blood Research Institute. She's also a lecturer of Immuno-Oncology at the IMC University of Applied Sciences in Krems, Austria. And in 2024, so just last year, Tina received the ASciNA Junior PI Award for her great work, the work she will discuss today. So the title of her talk, Advances in Cancer Immunotherapy, Orally Active Small Molecule Inhibitor of PTPN2 and 1, inflames the tumor microenvironment and unleashes potent NK and CD8 T cell immunity. The floor is yours, Tina.
Okay, thank you very much, Alexandra, and also thank you to the ASciNA for inviting me to discuss our work today. And I also want to again express really how deeply honored I am to have been awarded the Junior Principal Investigator Award last year. So thank you very much again. But let's jump right in to the topic, and I will share my screen. Everything looks well to you. So cancer immunotherapy has transformed patients' lives really significantly. This is compared to more traditional treatments like chemotherapy. It can lead to this long survival and in some patients even cure. However, not all patients respond to immunotherapy, leaving a big need for new and innovative therapeutic options. So today I would like to tell you the story how we discovered a new immunotherapy that is an orally active small molecule inhibitor of the phosphatases PTPN2 and N1, and that characterizing the mechanism of action of this inhibitor was truly thrilling to us because it completely remodels and inflames the tumor microenvironment by directly acting on tumor cells to make them more sensitive to immune attack, but also activating the immune system to unleash very potent NK and T cell anti-tumor immunity. So just as a way of background, cancer remains the second leading cause of death in the US, following the number one people killer that is and remains heart disease. And it has so for several years. And here you can see the statistics for 2023, but projections for 2024 are similarly dire, and it is the same in Europe and it's the same worldwide. So cancer really is the second leading cause of death. About up until the mid-1900s, cancer immunotherapy or cancer therapy was dominated by what we call cut, burn, and poison. That is surgery, radiation, and chemotherapy. However, more recently, towards the new millennium, targeted therapy and immunotherapy have gained a lot of attraction and has also shown some promising results. So immunotherapy specifically aims to activate or reactivate the cells of the patient's own immune system to recognize and kill tumor cells. But immunotherapy can also target the tumor cells so that they don't actively suppress an immune response, and so they can better be recognized by immune cells in order to be killed by them. So a little bit more about our immune system. It not only protects us against dangers, but also is responsible for maintaining healthy tissue via these really specialized cells of both the so-called innate, but also the adaptive immune system. So innate cells are very quick and non-specific, quick in their response and non-specific, involving cells like dendritic cells and NK cells or natural killer cells, and also macrophages. And then adaptive responses are a little bit slower, but they are also antigen specific and involve T and B cell responses. So the benefit of those cells the cells of the adaptive immune cells is that they can provide really long-term protection against recurring dangers through immunological memory. So they form memory cells. And for example, vaccination against pathogens like viruses really aim to induce such immunological memory so that you can be protected against a second viral infection later in life. And together, the adaptive and the innate immune cells eradicate pathogens. They protect against toxins. They can kill cancer cells, but also importantly, they're also required for tissue repair. So in order to do all this, immune responses are really highly regulated and have safeguards or checkpoints in place to prevent an overreaction, which otherwise can cause either autoimmunity or immunopathology that would induce real tissue or organ damage. So cancer cells, however, have exploited this pathway for immune evasion mechanisms. And here, the by now quite famous cancer immunity cycle proposed by Dan Chen and Ira Mellman provides a framework how to understand this series of events that generate an entire anti-cancer immune response. So it emphasizes this iterative nature of the circle where the death of immune cells by either therapy or by T-cell mediated killing initiates subsequent rounds of antigen presentation of tumor antigen by cells of the innate immune system, like dendritic cells. These DCs, dendritic cells or DCs is the abbreviation, DCs move then to the lymph node where they can activate T cells. The T cells then infiltrate back into the tumor, kill those tumor cells, and then those dying tumor cells restart this process again. So any step of the cycle can, of course, become rate limiting, and several suppressive mechanisms can be induced by the tumor, rendering the immune system unable to control tumor growth. And examples are here these T cell negative regulating molecules, CTLA4 and PD-1, which you may actually have heard of because targeting those with blocking antibodies has been very successful as cancer immunotherapies. And those are called immune checkpoint blockade or immune checkpoint therapy. The goal of immunotherapy of this checkpoint blockade specifically is to reinvigorate T cells. Both CTLA4 and PD-1 are expressed on T cells. And when they engage by the ligands that are expressed on antigen-presenting cells or tumor cells, can mediate negative signals into the T cells, suppressing their function, so that the T cells then cannot kill a tumor cell at all anymore. And so I do want to emphasize again, though, that these mechanisms do have a purpose, that is to prevent T cells really from overactivating and overreacting and then acting too long, too strong, and then cause harm to tissues. However, in the tumor, we want to, of course, reach the highest possible and strongest T cell response that we can get. But tumors are quite smart, and they often upregulate this PD-L1 as an immune evasion mechanism, so that, again, they kind of enable the T cells to kill them. So what scientists, however, about 15, 20 years ago now, have discovered was that blocking those interactions between PD-1 and PD-L1 or CTLA4 and its ligands, that can really prevent the T cell inhibitory effect of those checkpoints, and with that then unleash the regular T cell function and restore this tumor killing activity. And for the discovery of such checkpoints, and their function, Jim Allison and Tezuko Honcho, actually received the Nobel Prize in 2018. And it was not for the discovery of the immune checkpoint blockade, it was really for the discovery of those checkpoints, the molecules themselves, PD-1 and CTLA-4. Now, immune checkpoint therapy with either PD-1 or CTLA-4 blockade is now really widely used in many different cancer types in the clinic and has truly revolutionized cancer care. One of the keynote trials shown here demonstrates this transformative success in which an anti-PD-1 antibody called pembrolizumab significantly increased the progression-free survival of non-small cell lung cancer patients over the chemotherapeutic arm here shown in gray. However, as you can tell, there is a significant unmet need for patients who, at some point start to progress on checkpoint blockade, or they're even resistant here to therapy to begin with. And in order to help those patients, we clearly need to better understand those resistance mechanisms, and we also then need to discover novel immunotherapy approaches to overcome this really high unmet need that remains here. So scientists at the Broad Institute in Boston, including Rob Magusa and Kathleen Yates, set out to perform an in vivo CRISPR gene knockout screen in mice to identify genes in tumor cells that, when knocking out those genes, make the tumors that are originally resistant to immune checkpoint blockade now sensitive to this therapy. And they got several hits. And then when they took a closer look at the top hits shown here, and you will also see here, this is a frequency plot on the left side here, they found that PTPN2 single guide RNA, so each gene was targeted by 4 guides, so therefore you can see these 4 red lines, that those were significantly depleted from the tumors as compared to average guide RNAs. And you will see that here in this average guide distribution in gray, as well as in the histogram on top here. So then Rob teams went further to characterize PTPN2 specifically, and they individually knocked it out of B16F10 tumor cells. And they validated that PTPN2 knockout, shown here in the red line, sensitizes tumor cells to immunotherapy, as demonstrated by a significant growth inhibition compared to the control tumors treated with just immunotherapy that was anti-PD-1 and an immune cell booster, GVAX, just by itself. And that's shown here in this gray line. So why does PTPN2 sensitize tumor cells, though, to immunotherapy? And so PTPN2 and also its paralog PTPN1 are tyrosine phosphatases that negatively regulate multiple different signaling pathways in a variety of different cell types. And here shown in tumor cells, they target kinases, including receptor tyrosine kinase, Sark family kinases, JAK and STAC proteins. And so what Rob and colleagues found was that both PTPN2 and 1 negatively regulated interferon signaling in tumor cells, and that when they knocked either one out, it promoted interferon signaling, regulating this pathway, increasing the response to it, and resulting in increased transcription of interferon response genes that then led to downstream effects that are really beneficial in the tumor microenvironment, including inhibition of tumor cell growth, increased immune cell attracted chemokine production, as well as increased antigen presentation. And we'll talk about that a little bit more in the coming slides. But then additionally, there was also published evidence that PTPN2 and N1 are also negative regulator in a variety of immune cells, downstream of cytokine signaling, and also in T cells downstream of the T cell receptor. And that was by targeting JAK-STAT signaling as well as FIN and LCK. And in line with this, genome-wide association studies associated single nucleotide polymorphisms or SNPs in PTPN2 and also one PTPN1 with autoimmune disorders. And that knockout of those phosphatases in a variety of immune cells increase their activation and effector function as shown on the left side here. So PTPN2 knockout in T cells drives increased activation and proliferation, cytokine function as well as effector function. It does so through promoting the T cell receptor signaling as well as cytokine signaling. So, and now is really where the beauty and the exciting part of large collaborations across academia and industry comes in. So we have this wonderful and extremely fruitful collaboration among the Broad Institute. We work very closely with Rob Mangusa's team, who I just mentioned before on the in vivo screen, with Calico and with Calico Life Sciences in South San Francisco, and then us, of course, at AbbVie. So we could rapidly, what that led to is that we could very rapidly ID as well as validate PTPN2 and N1 as targets for immunotherapy, and then also quickly initiate efforts on the discovery of PTPN2 and N1 small molecule inhibitors. And while this was very promising from the biology side of things, there was a ginormous hurdle to overcome, and I should say for our chemistry team here really. To inhibit the function of a protein, one ideally targets the catalytically active site. But in the case of a phosphatase, the active site is highly polar. So the basic region that encompasses the active site is shown here in blue in this figure. And molecules that bind to the site really historically have very poor cell permeability and therefore poor cell activity, of course. They also have poor drug-like properties and are not orally available as just a pill to swallow. So currently there have been no approved drugs that have emerged from either PTPN2 or N1 efforts over the last 10 to 20 years even. And the overall conclusion in the field, as a matter of fact, was that the active site of phosphatases are just undruggable and nobody continued trying it. However, for our efforts, we aimed for a dual PTPN2 and 1B inhibitor, as our hypothesis was that targeting both of those phosphatases would increase anti-tumor activity over each individual phosphatase. And PTPN2 and 1 have high homology in this active site here. And additionally, there was no allosteric site for PTPN2 that has been identified. So we really had to focus on the active site here to design our inhibitors. And ultimately, our truly outstanding chemistry team here, and two of the wonderful chemists are on the call today, Jennifer and Xiaoming, indeed succeeded and discovered ABV CLS484, which I will refer to in the following just as 484. And this molecule makes several key interactions and is shown here in green bound to the active site. And it has a single-digit nanomolar potency and is highly selective for PTPN2 and N1 and has no activity on some of the other phosphatases that you may have heard of, SHP1 or SHP2. And I really cannot call out enough the Herculean effort of our world-class medicinal chemistry here, led by Jennifer Frost on the call, enough that they really have made the impossible possible and drugged the undruggable. So a really great effort here. But so with this potent compound 484 in hand, we wanted to, of course, characterize it further. And we first asked the question if pharmacologic inhibition of PTPN2 and N1 with our inhibitor resembles the phenotype of the genetic knockout in tumor cells that has been reported in the past, in that it increases the responsiveness of the tumor cells to interferon. And the answer was yes, it did. And we found several effects really beneficial in the tumor microenvironment. So 484 dose-dependently enhanced interferon-induced growth inhibition in B16 tumor cells, and similarly enhanced antigen presentation on tumor cells, here shown as an increase in the ovalbumin immunodominant epitopes in fecal presented on ovalbumin-expressing B16F10 cells. So these effects are specific to interferon treatment, as 484 does not have any impact on untreated tumor cells, shown here in the white circles. And then in co-cultures with T cells, we observed that this increase in antigen presentation observed here on the tumor cells also resulted in increased T cell mediated killing of those tumor cells, indicating that 484 treated tumor cells could be better recognized by T cells and therefore better killed. And then finally, also very interesting to us, 484 elevated the production of interferon-induced chemokines from the tumor cells, so that more immune cells could be attracted into the tumor. And then, in addition to its effect on tumor cells, 484 also impacts immune cells and promotes their activation on function, in line with what has been reported for the PTPN2 and PTPN1 knockout immune cells that showed really enhanced activity of these T cells. So what we did is we stimulated splenic T cells through the T cell receptor and treated on top of that with 484. And that demonstrated a dose-dependent increase of the activation marker CD69, shown here in orange, and also to a dose-dependent production in pro-inflammatory cytokine, exemplified here by interferon gamma in green. But we also have data on other pro-inflammatory cytokines. And then, very importantly, 484 did not activate unstimulated T cells, as you can see here. Rather, it enhanced the function of already activated T cells. And those were extremely good news to us, because we really don't want to unspecifically activate naive T cells in patient circulation. And then we were very happy to get those results, as here this confirms our hypothesis that our dual PTPN2 and 1 inhibitor showed greater benefit than targeting one of the phosphatases alone. So what Pat Truss did, is she knocked out either PTPN2 or N1, and then stimulated T cells via the TCR with or without 484 to see if it would further enhance the T cell activation of already the knockout T cells. And she measured that then by CD69 expression on those T cells. And then, as expected, both single knockouts increased T cell activity, and you can see that here, compared to the control in gray. But additionally, and this was exciting, 484 dose-dependently boosted this activation in the single knockouts compared here to the untreated T cells. And this really also highlighted again that it has this further amplifying function, and that targeting both phosphatases and 2 and 1 was really, really critical. And then again, very importantly, neither the knockouts nor the compound in itself activated unstimulated T cells. So with these encouraging in vitro data, then we assessed the efficacy of 484 in vivo. Oral dosing of 484 demonstrated monotherapy efficacy in anti-PD-1 responsive models, including the MC38 model showing here, in which both anti-PD-1 in blue as well as 484 in red led to tumor regression, as well as prolonged survival, as seen over here in the Kaplan-Meier plots, which also included cures, exciting. And then additionally, in anti-PD-1 resistant models, the 41 tumor model here, in which the tumor actually does grow out with anti-PD-1 therapy, here in blue, but now 484 demonstrates significant efficacy and survival advantage as well. And then in the models that are sensitive to anti-PD-1, we find that 484 leads to much, much deeper responses in combination with PD-1. So in the MC38 model, again, PD-1 and anti-PD-1 and 484 monotherapy actually each provide maximum efficacy. So what we had to do to evaluate combination potential is use suboptimal dose levels. And as shown in the blue anti-PD-1 for anti-PD-1, as well as in green for 484. Yes, these suboptimal doses slowed down tumor growth. However, a combination of the both led to tumor regression and survival advantage, very similar to the full doses of 484. And we find actually the same results in yet another model, the CT26 model, in which now full doses of anti-PD-1 and 484 induced significant tumor growth reduction. But again, the combination of both individual monotherapies provided much, much deeper responses and prolonged survival, and also even actually included cures, which is pretty exciting. So, and this was very thrilling to us, detailed investigation of the tumor microenvironment by single-cell RNA-seq analysis. And here shown is the data from the KPC model. This is data from our colleagues at Broad, but we have seen very similar data in, across different syngeneic tumor models, also across the collaboration, all three sites demonstrated that 484 completely remodeled the intratumoral landscape in very different ways than anti-PD-1 actually does, which could help explain that those therapies demonstrated this combination efficacy because they just target different mechanism of actions. So if you want to focus on the yellow boxes here, 484 increases the T cell and NK cell abundance in the tumors and induced also fewer of the immune suppressive M2 macrophages, while it promoted the anti-tumorigenic M1 macrophages. And while in the interest of time, I really can't show you all the data, but we performed in vitro experiments on isolated NK cells, macrophages, and actually also dendritic cells. And we find that 484 had direct effect on each individual immune cell type and induced in each of those pro-inflammatory anti-tumor activity, which was nice to see that we're impacting various different immune cell types. So we then performed multi-parameter immune phenotyping by flow cytometry of the tumors and found that while anti-PD-1 and 484 induced higher frequency of granzyme B producing T cells than the vehicle, with granzyme B being cytolytic protein that induces lysis of target cells, looking at the ratio of the non-regulatory T cells, so FOXP3 negative cells, to FOXP3 positive cells, which indicate the regulatory T cells, so suppressive T cells, 484 clearly shifted this balance to its lower levels of the suppressive population and more towards effector cells capable of tumor cell killing. Additionally, we found a higher frequency of NK cells, which can also directly kill tumor cells. However, anti-PD-1 did not promote this NK cell type. And we were particularly excited about this, to see this NK cell effect, because a role for PTPN2 and 1 in NK cells has not been described ever before. And Katie McGuire, here listed, really led all these efforts and described a lot of the effects further, which you can find in our paper. And we wanted to focus more on what the functional impact of 484 on T cells is, because both the single-cell RNA-seq data as well as immunophenotyping indicated that 484 has very strong effects on T cells, which seem to be quite different to anti-PD-1. So we wanted to know what's going on. So a big problem in the tumor microenvironment that prevents T cells from killing tumor cells is a phenomenon called T cell exhaustion. And this is a state that T cells acquire and results in T cell dysfunction when they just are long-term exposed to antigen. And this often happens during chronic infections like HIV or HPV infection, and also in cancer. And so T cells then get progressively exhausted the longer they see this cognate antigen, with this final stage called terminally exhaustion. And with terminally exhausted cells, those cells, they then express high levels of a surface marker called TIM3, as well as high levels of a transcription factor called TOX. And the gene signature that has been established for those cells in the clinic correlates with very poor responses and prognosis for immune checkpoint therapy. And then similarly, as listed here, in preclinical mouse models, the response to immune checkpoint is so that it only transiently reinvigorates exhausted T cells and mostly has effects on cells early in the exhaustion process rather than these terminally exhausted cells. So what we then excitedly found is that with 484, we significantly reduce this terminally exhausted population, this TIM3 and TOX double positive population in tumors of mice. And not only did it reduce the frequency of these cells, but also on a per cell basis, it reduced the level of expression of TIM3 and TOX, as shown here by the mean fluorescence intensity. And this is exciting to us because this is different to a previous study from Marty Lafleur in Arlene Sharp's lab, in which they showed that PTPN2 knockout in T cells, so again, just PTPN2, but also knockout, so complete, knockout of the gene, not just inhibition, enhances the formation of this terminally exhausted T cells. So pharmacologic inhibition with dual inhibitor had clearly different effects here. And so our colleague at Broad then sorted those exhausted TIM3 positive cells from tumors, and they performed ATAC-seq to assess chromatin accessibility in those cells. And indeed, looking here at TOX, as an example of the exhaustion-associated genes, 484 reduces the chromatin accessibility here compared to the non-targeted control, which results in just lower transcriptional activity compared to PD-1 and also the control. And then excitedly, at the same time, we find more chromatin accessibility of genes that are associated with this long-term memory formation and long-lived cells, including IL-7 receptor, shown here on the right side, is really what we want for anti-tumor immunity. And you can see here, more accessible chromatin with 484 compared to the non-targeting control or anti-PD-1. And then to confirm that 484 indeed directly lowers exhaustion of T cells, we performed an in vitro experiment developed by Rebecca Matthew and Kerry Backus, in which we chronically stimulated T cells in 5 rounds with antigen. And then starting from the third stimulation here, we added 484 to these cultures. And then at the end, we read out the frequency of the terminally exhausted cells that we found. And so after 5 rounds of stimulation, 484 compared to the vehicle control here dramatically reduced the frequency of exhausted T cells. And here we're looking at this PD-1, TOX1 double positive population. And at the same time, 484 increases the multifunctionality of those cells, indicated by cells that can produce both interferon and TNF at the same time. And loss of this multifunctionality really is, by the way, a hallmark of T cell exhaustion. So this was wonderful to see. And then when we then took those cells and co-cultured them, so these T cells, right, and co-cultured them with tumor cells expressing the cognate antigen and monitored the viability of the tumor cells over time, we find that indeed the tumor killing function of these exhausted cells was rescued by 484. So here in purple, you can see the tumor cell growth just without T cells, and the tumors grow out very quickly. If you just add one-time stimulated T cells, so the good and strong effector cells, those kill tumor cells really well, and that is shown here in the black line. So exhausted T cells in gray here are the 5 times stimulated cells, and those have a quite hard time to controlling tumor growth. But if we add now 484 towards the end of the culture, right, so these cells are already exhausted, we can then rescue some of the tumor killing function of those T cells. And you can also see this here in this cytotoxic graph on the right-hand side. So now I would come to a part that is relatively new. So you will see, and we're quite interested in working on that right now. So you're the first people to see this data. And I will just give you a teaser of 2 data slides. So you will have to wait for upcoming presentation to find out a little bit more. But with this really intriguing data in T cells, that following 484 treatment, the T cells become more activated, they produce more cytokines, are more cytotoxic. I didn't show you that, but they're also metabolically way more active. And then additionally, they are less exhausted. We ask if 484 could improve CAR T cell therapy that still faces significant challenges. So we did see transformative impact of CAR T cell therapy in the clinic in cancer treatment, but there are really major significant hurdles, particularly for the treatment of solid tumors. So there is an unmet need, and we can hopefully improve that. In general, CAR T cells are T cells that are extracted from patients' peripheral blood, as you can see here in this little schematic, and in vitro engineered then so that they express a chimeric antigen receptor, abbreviated CAR, that then therefore CAR T cell therapy, that targets a cell surface protein on tumor cells so that the T cells can now specifically kill those tumor cells. And CAR T cells have really revolutionized the treatment options for hematologic cancers, resulting in long-term remission seen in patients, as you may be familiar, this very famous case of Emily Whitehead, who was the very first pediatric patient receiving CAR T cell therapy and who remains cancer-free now many, many years after she received her first treatment. So now there are 6 FDA-approved CAR T cell therapies showing significant improvement in the overall response rate in hematological indications, like showing here, for example, for multiple myeloma, as well as diffuse large B-cell lymphoma. However, despite these successes, many challenges remain, particularly, like I mentioned, in the solid tumor space, where CAR T cells need to really infiltrate this often very hostile and immune suppressive environment of the tumor, and with issues like CAR T cell persistence, and then also importantly, the aforementioned T cell exhaustion. So we really wanted to see if our inhibitor, theoretically alleviating all these issues, could improve the efficacy of CAR T cells against solid tumors. So again, this data not shown before, so please don't post it anywhere, and a ton of data coming up soon. But our colleagues at Calico, Chirag and Meng, developed a mouse CAR T cell model in which they generated B16F10 tumor cells that expressed an endogenous antigen. In this case, it was human CD19. That then can be recognized by CAR T cells that, again, they specifically engineered to recognize this CD19. So after they inoculated the tumor cells and those formed solid tumors, these mice were treated with a lymphodepleting agent, as is done in the clinic, to create space before transferring the new CAR T cells. So it creates space for the new CAR T cells to really expand in the patients. And we here for these experiments use cyclophosphamide. And we compared then mice that we treated either with vehicle or with our inhibitor, or with CAR T cell therapy, or with a combination of CAR T cell plus our inhibitor. And then we monitored the growth of these solid tumors. So the first exciting thing to see was that our inhibitor in red here promoted anti-tumor immune response despite this lymphodepleting therapy compared to the vehicle. So that was quite interesting. The vehicle is in black here. And as for the CAR T cell, Chirag and Meng actually transferred here a very low number of CAR T cells to only weak, so here in blue, to only see a weak response to the CAR T cell therapy, as is often seen in the clinic when the therapy does not appear to be very effective. So this is actually the situation we would want to improve in the clinic. But now, excitingly, adding our inhibitor to the suboptimal CAR T-cell therapy induces much stronger anti-tumor responses and tumor regression. So this was really interesting to see. And we were also very happy that not only was there high frequency of CAR T-cells in the tumors when adding our inhibitor, but the CAR T-cell population was also more cytotoxic, as indicated by high frequency of coenzyme B producing cells and perforin double producing CAR T-cells. And additionally, as we really hoped for, these CAR T-cells had a much less exhausted phenotype. So coming back to the cancer immunity cycle, and I just have 2 more slides to go, while currently approved immunotherapy, anti-CTLA-4 and PD-1 checkpoint blockade, here shown in blue, those mostly act at single steps of this cancer immunity cycle. 484 acts at multiple steps of this cycle. 484 increases dendritic cell activation and antigen presentation. It augments T cell activation and chemokine production from tumor cells to direct those T cells into the tumor. It induces a pro-inflammatory tumor microenvironment, more amenable to anti-tumor immunity by shifting macrophages towards this pro-inflammatory properties. And then by acting directly on the tumor cells, 484 increases their antigen presenting, leading to better recognition and killing by the T cells. And it also amplifies really a growth arrest, so intrinsic to the tumor cells of the tumor cells. And then finally, T cells and NK cells in the tumor are much more cytotoxic, so are better cancer killers. And these T cells are also less exhausted. So we can see that 484 acts at multiple different ways to increase anti-tumor immunity and tumor killing. So a quick summary, what I would love for you to take away from this talk is that 484 is a first-in-class oral PTPN2 and N1 dual inhibitor that drives very robust anti-tumor immunity. And it does so by, number one, sensitizing tumor cells, so directly acting on tumor cells and sensitizing them to immune-mediated killing, but also directly acting on a variety of immune cells and promoting their activation and function, including the actual tumor killers, NK and T cells. And as for T cells, we had a couple of very exciting observations here. Something I didn't show you, we found that our inhibitor increases the diversity of the T cell repertoire. So now we can tackle the tumor on multiple different sites. It also enhances their metabolic fitness, increases effector function, reduces T cell exhaustion, and now we also have data showing that it promotes the effectiveness of CAR T cell in solid tumors. And then also a very important point, it has a very different mechanism of action than anti-PD-1 treatment has. And it also, and likely therefore, also works in combination with anti-PD-1 really well. And 484 is currently tested in patients with solid tumors in a phase 1 clinical trial. So I also don't want to miss out on showing you some of the faces at AbbVie who greatly contributed to the project, and also show you that while, as in every project, right, we had a lot of challenges to overcome, but one, our fearless project director, Phil Kim, navigated us through all of them. And then second, we also had many, many rewarding moments. One of them is captured here from 2020, when we were able to watch the very first capsule filled to be dosed in cancer patients that came up off the production line in our manufacturing building directly here on campus. And we all watched this really goosebump moment of capsule filling through a tiny observation window, and our pharmaceutical colleagues in U showed us this really very first capsule close up. So of course, absolutely unforgettable moments here. And of course, I can't resist a little promotion of our paper to increase our access hit. So please scan that and look through it. There's much data than I could possibly talk about to you today. And very importantly, can't emphasize enough the great teamwork across a large team at AbbVie and our colleagues at Calico and the Broad Institute that really led to drugging the undruggable. And I also wanted to show you some of the key folks here from Calico and the Broad who were extremely instrumental, helpful, and great collaborators. And you can see it truly took a village of extremely dedicated scientists. So I wanted to thank them all again for being a wonderful team, and then you for listening. And I'm happy to take any questions. And maybe I'll stop sharing.
We see you more. I have the first question. What are the next steps now?
Yeah, so number one, as I mentioned, the clinical trial is ongoing, of course. So we're testing it both as a monotherapy and in combination with PD-1 in solid tumor indications. But in parallel, we're investigating a lot where PTPN2 can also act and what potential other combinations where we see, like, for example, with CAR T cells, where we see there is room for improvement in the clinic on whether our inhibitor could make any of those additional therapies more effective here. So we still have a lot of experimental questions to address here, and it's exciting times for the program.
Bernhard Gruber has a question.
Yeah. Hi, thank you very much for the good talk. It was indeed a challenge to follow a bit because there were so many dedicated words in there that I'm kind of a little bit familiar, but I'm coming from a different field, so from magnetic resonance imaging. And what I was questioning is in the field of oncology, where does it exactly fit in? We know in clinic there's several combinations of treatments, and how does this all fit into the oncology for you?
Yeah.
What is the potential?
The way we view therapeutics in oncology is that, right, a lot of the therapies that we currently have that includes chemotherapy, radiation, even targeted therapies with ADCs, they're often just temporal responses, right? We see great tumor regression initially and early on as many patients respond, but most of them will progress. Where we see the big potential with immunotherapy, including our drug, is because we're activating the immune system, we're inducing this long-term memory so that when a tumor, right, we can now, with what we call these debulking agents, right, that can basically reduce the mass of the tumor, we can then come in with the Immuno-Oncology therapies, induce this long-term memory that now we can protect these people from recurrence of the tumors and potentially cure them. So we are living really by, right now the field thinks about it, that we have to do this double hit, right? This debulking and inflaming. So it's a combination of more traditional and standard therapy, but also not so traditional, right? These targeted therapies like ADCs or some T-cell engager, right? So this debulking, but then coming in with IO and really driving this long-term responses, because that is what chemo, radiation, ADCs cannot do for patients. So that's what we're hoping for.
Okay, so maybe as a follow-up question, so do you view it more as if you had cancer and you don't want to get a reminiscence or whatever, that the likelihood of this cancer showing up again, so this drug can actually help to, if you take it over several years, which the clinical studies are going to reveal, that this can suppress or support basically the body in fighting the cancer to popping up again?
Yeah, so what we're actually thinking is that even with a shorter course, so not necessarily over years and years and years, but maybe a couple months or so of treatment, then I will say that we're figuring out exactly how to use this drug in the clinic as we speak, of course. But that could induce this memory pool of immune cells that protects you later in life, right? So once you have memory cells and then they can attack your tumor, you don't necessarily need the drug anymore to later, if years later, you will see the drug, it can respond. It's very similar. You can imagine it very similar to just regular vaccination, right? You give the vaccine first, and then you form this memory, and that can protect you later in life. Yes. But that's exactly the idea, like you said. Exactly.
I wish you all the best for the clinical trials, and I hope you basically found a vaccine for cancer, or for certain types at least.
Thank you. Yes, it's not. I have to say though, very clearly, it's not a vaccine.
Yeah.
And why I'm saying that is what you mentioned is because cancer vaccines right now are super hot. There are new findings in the field right now.
It sounds sexy, but it isn't.
Yeah. But there are cancer vaccines now. Yeah.
Okay, let me correct that. So a vaccine type kind of drug. So that may be that.
An immune stimulator. Anyway. Let's call it that.
Immune stimulator. Okay, let's agree on that. Thank you very much. Thank you very much, Christina.
Okay, thanks for the question. Good question. Yeah, Matt.
I want to.
I don't know how to use the raise hand function, so
that is good.
Yeah. So the dual impact on the immune cells and tumor cells is very interesting because it can do different things for different tumor types. And so I was wondering if you could comment on the diversity of tumor types and whether the impact on the tumor itself or the immune cells are, more or less important in different tumor types, and then whether that translates if you've looked into CNS metastases or CNS tumors.
Yeah. So excellent questions, but many questions in one question. But so yes, in the clinic, right, we don't know. We're figuring this out. Preclinically, I can tell you that we have seen responses in, what sometimes people recall is colder tumors. So in hot tumors, it works fine. And those are the highly inflamed tumors where you already have your T cells, right? You have an immune response going on. PD-1 works super well there. But we have interestingly also seen responses in mouse models of colder tumors where we have lower inflammation, right? We have a lot of these myeloid suppressor cells that act through a different mechanism to suppress the entire anti-tumor immunity, where anti-PD-1 does nothing for the mice, right? We have seen responses with our inhibitor there as a monotherapy in itself. So we don't need even anything on top of that. So we're hopeful in the clinic that we can, target a variety of tumor cells. On top of that, I will say we did large tumor cell screens just in vitro, right? And we treated those on top of that with interferon gamma because hopefully it was clear that without interferon gamma, without a stimulating signal, our inhibitor, the cells couldn't care less about it. This is great news, right? Because it also alleviates some of the potential tox implications that you could think of. But so we have done that, and we also see a variety of different cancer types responding to that. So yes, there is a tumor intrinsic effect, but it depends on the in vivo cancer model. And based on your question, I assume that you are in the field and that you know that all the in vivo cancer models are a little bit different. So in some of those cancer models, we do find that when we knock out PTPN2 and N1 in the tumor cells, we still get a very similar response, indicating that it's really the immune system, the major driver of killing those cells. That doesn't mean that there couldn't be an additive effect in certain tumors, right, that you also see through this interferon effect for sure. Your other question, just quickly wanted to add on the metastasis because we thought that was fascinating. You can find that also in our paper. Metastases are mostly controlled by NK cells. So excitingly, we found that we also had the effect on NK cells. And what we noticed is we actually ran a couple of metastasis models. One was a tumor seeding model, but then the other one was an actual metastasis model where we implanted a tumor, a breast cancer tumor, and then we looked at the metastasis to the lungs. And we also find that our inhibitor dramatically reduces this metastasis, and it does so then in an NK cell dependent manner. So CD8 cells don't play much role, and it just perfectly fits so that the hypothesis that we have about, or what is known in the field about metastasis, and now really is where the NK cells kick in, right? So it's this very nice, dual mechanism again, where in a scenario where the T cells cannot work, and this is very often tumors, again, I mentioned these are really, really smart, little bastards. They develop immune evasion mechanisms by also downregulating molecules that T cells, however, require to recognize the tumor. And these are things like, for example, major histocompatibility complexes, so MHC molecules. This is JAK-STAT signaling, which upregulates MHC. So if there's no MHC, T cells can't do anything against CD8 cells. In that case, though, really the NK cells kick in and can kill the tumor cells. So we've seen it really beautifully in vitro as well as in vivo. Yeah, great question.
Yeah, thank you for that. It's just by the logic, it should also work in the CNS. Have you seen that?
I'm so happy our medicinal chemist Jennifer Frost is there. To maybe help answer this question about brain penetration. We haven't looked at, if the question just is, did we look at brain tumor models? We have not, but I will defer a little bit if you don't mind.
No, so right, these compounds are really polar, right? So we don't expect large amounts of compound in the brain. So I would probably not expect that, but I think Tina alluded to creating a memory effect in the body. So in theory, down the road maybe, but not right now.
Perfect. Yeah, and we also see, by the way, if you're talking about brain mets, right, we do see a abscopal effect. So meaning, right, if we're treating, if we're treating a primary tumor, at a site, maybe in a flank, right, we also see that we get responses throughout the body if we inject a different tumor somewhere else in the body. So, and that again indicates that we have memory formation. I would also say some of the brain tumor models, you're breaking the blood-brain barrier, just the way the tumors grow. So in that case, maybe it could, but yeah.
Wonderful. Thank you so much, both.
Thank you. Yeah, good questions. Thank you.
Everybody, we have time for one more question. And you can show us your face too.
We don't want to scare anybody away, right?
Please do, though. We're not scared.
Tina, if you want, put up the QR code again for the paper.
I do, yes, want to put up the QR code for the paper again.
And also, everybody, we will post the video on the ASciNA YouTube channel. Once Tina has given us permission to do so.
I give you the permission now. But here, this is Jennifer Frost leading our medicinal chemistry. Again, drugging the undruggable. Here, Xiaoming is a chemist on the team who developed our second clinical candidate. And then this is our fearless leader here. So.
Well, everybody.
Okay.
That's a great team. Thank you for this amazing work. Thank you for the great presentation. Thank you for your collaborators to be on this talk as well. It's great to have them.
Yeah, thanks guys for joining. Really appreciate that you dialed in.
So Tina, thank you. Really, congratulations again. This is amazing work, and we look forward to hearing how it progresses. We can schedule another one, a follow-up in a year or so.
Happy to do that, always. Okay. Thank you so much for the invitation, Alexandra. I really appreciate it.
Absolutely. Thank you all for joining us.
Yeah.
That's recap it, and everybody have a wonderful rest of your day.
Thank you.
Thank you so much. Bye, you guys. Thanks for the questions.
Bye. Bye-bye.